Hello, my name is Maria Sanchez Garcia. I'm going to present the study Lost in Transition under investigated mechanisms linking organic and inorganic carbon pools in calcareous soil. This research is being developed by the people of the Department of Soil and Water Conservation and Organic Waste Management of the Spanish Research Council. If you want to know more about our group or our research work, you can also click on the link below. The interactions between soil organic and inorganic carbon pools have been scarcely investigated. However, this process can be of paramount relevance in arid and semi-arid soils, where inorganic carbon prevails and represents one of the most important carbon reservoirs in terrestrial ecosystems. For example, organic carbon can be transformed into the inorganic carbon pool through the release and subsequent precipitation of secondary carbonates, which could constitute a pathway to inorganic carbon sequestration in the soil. On the other hand, inorganic carbon in the soil can be lost through the solution of carbonates and the release of carbon dioxide to the atmosphere. The fate of carbon dioxide will depend on many factors, such as the soil pH, moisture, or calcium availability. Our objective was to study the effect of common root exudates on the interactions between organic and inorganic carbon species within a calcareous soil matrix. Several carbon-13 labeled substrates were added to a carbonate-rich soil in laboratory incubation experiments. Glucose, acetic acid, and proline were the sources of labeled carbon added to the soil in 3 consecutive drying-rewetting cycles. During the 36 days of soil incubation, carbon dioxide fluxes were monitored. At the end, we measured organic and inorganic carbon in soil solution as well as in the bog soil. Isotopic signature in all these carbon pools was also measured. All carbon additions were associated to a peak in carbon dioxide fluxes. Cumulative carbon dioxide emitted was similar, and the differences in flux dynamics were associated to the carbon source. Carbon-13 signature was measured in each of these flux peaks, and the differences between treatments were significant. Proline was the treatment with the highest delta 13 after all carbon additions, followed by acetic acid, and finally by glucose. Although cumulative emissions were similar between treatments, the highest proportion of emitted carbon from the labeled carbon source was derived from proline addition, whereas the lowest proportion corresponded to the carbon derived from glucose addition. On the other hand, labeled carbon-13 derived from proline was the lowest of the inorganic carbon fraction of the soil solution at the end of the incubation, whereas it was the highest in the case of acetic acid. The induced changes in soil pH probably affected the carbon-13 signature of the inorganic carbon of the gaseous and the liquid fractions. Carbon-13 signature of the bog soil was also differently affected by the carbon source. Consistently, carbon-13 signature of the soil organic carbon increased in all treatments, which was much more evident in the glucose and acetic acid treatments. Carbon-13 signature also increased in the case of the inorganic carbon fraction. Our results show that a fraction of the added labeled carbon was incorporated to the soil inorganic carbon pool. Consequently, estimations of soil carbon sequestration or priming effects should account for the mechanisms linking organic and inorganic carbon pools in calcareous soils.